Welcome you all to this Cryo-EM course. Last class we discussed about how C1 lens is working, that okay, if we have a very strong magnetic field, how it is going to affect our electron beam, as well as how the crossover point, means that converging point, it is changing its position. Now we are using that parameter, that okay, there is a C1 lens current changing and crossover point it is changing, as well as, so C1 lens current, if we just remember that C1 lens basically it is regulating how much electron rays are going towards the C2 lens, right? If this is our C1 lens, it is regulating that how much electron it is going to the C2 lens, and then it is considering how much electron it is going towards my objective lens. So if we just consider that okay, there is a C1 lens and there is a C2 lens. So last class I discussed about only the C1 lens, that okay, there will be a crossover point, it is changing. If this is the crossover point from the C1 lens, what is the changes between the position of this crossover point. But if you remember, if you just look at this diagram, you can see all the cases that C1 lens is regulating how much electron dose is going through the C2 lens. So now we'll do the same diagram again. So we have electron and there is a crossover point is somewhere here, right? And then as I mentioned, there will be a C2 lens and then it will measure the crossover point here, right? So this one we can consider C1 and this one we can consider the C2. Okay, there is a other point where electron can converge little bit up. When electron will converge little bit up, it can go spread at the beam when it is crossing that C2 lens. Then C2 lens, we are not changing the magnetic field at all. Here one lens system we are changing the magnetic field, and that is the C1. The magnetic field I am changing the C1 only. C2 is a fixed at this moment. Now if it is fixed, definitely this electron beam is spreaded than the black ray, and the blue is more spreaded than black. Then definitely if we have the same magnetic field for the C2 lens, the converging point is going to change. There is no doubt about this. So it will change like that. Now if we have another ray, you consider that there is a red. So red ray it will come like that, spreaded more, and if it is going to converge, it is going to converge below the plane. One more possibility, that okay, I am using this pink color. If beams are converged something like that, then it is spreaded, right? And then this beam will converge something like that. So now you can see that okay, because of this fixed C2 lens, there is a change, and then rays are going in a different direction and beam diameters are changing. But here this crossover point, it is also going up. Now let's do one exercise, that okay, we have a fixed C2 lens and we'll try to converge all the electron beam in one particular point. We will not allow the electron beam to go and converge wherever you want. So we'll change the C2 lens current also in such a way that all the electron beam, it will converge in one particular point, and then it will spread from there. So if we just use this, here we have, this is our C1 lens, this is my C2 lens, and let us change both of them and then see what we can get it. So rays are coming here the first crossover point, then it is going through the C2 lens, and now fix this particular point, that it doesn't matter, we will use a magnetic field in such a way that all the rays will go through this point. So, we are changing C1 lens current and C2 lens current simultaneously in this particular diagram, but this point is fixed here. Now, let the rays are going and giving us an image. Now, there is a second ray. So, if there is a crossover point here, it will spread the beam, and definitely when there is a beam is spreaded, we need some extra care here to use that C2 lens current. We have to increase that C2 lens current, so rays can go through this particular center. So, if we just consider, it is going through this one. Okay. Now, I am using another third ray. So, they are converged here, and then it will go through this one, and now if we have the other possibility that okay, one ray is coming something like that, okay, and it is going through this particular point and giving us an image. So, all the cases you can see, even there is a fixed point, it will be, there will be some change in the beam diameter. It is not that beam diameter will be a fixed, but it is not going to affect like that. Here basically it is looking like that there is no control. So, this is not a very good setup where you have, we are only changing the C1. Here this diagram, it is showing that if we are only changing the C1 and C2, it is just the fixed one. So, what kind of beam diameter we can get? But here you can see there is a systematic way it is going, that okay, if we have a particular spot, that okay, this particular case your spot size is very small. The spot size is, you can consider 1. This black one, the spot size you can consider it is 3. This one you can consider it is 6, and this one you can consider that spot size is 8 or 9. So, you can see whenever we are going from very low spot size to high spot size, your due to very strong magnetic field, your beam, the crossover point, it is going up, and beam is spreaded too much in this particular region when it is going through the C2 lens. So, our C2 lens is somewhere here, right? When our C2 lens is somewhere here and we are using, beam is really spreaded when we are using the spot size 8. And then if we are changing this magnetic field of the C2 lens, we can converge this beam in one particular point, and it can go from one particular point, and it can give us a particular beam diameter. Now, in same setup, that we are going to use a high spot size and then certain intensity magnetic field, and will get a particular electron beam. Now, I am bringing aperture in this picture, that okay, if we have an aperture, how it is going to behave. So, we have electron, as I mentioned previously, there will be a crossover point, electron will go like that, and it will merge one particular point. So, it will be better if we just draw this diagram in a little bit different way. So, it will help us to explain. Now, it is going and giving us a point. Now, there is another possibility that okay, there is this one, then the electron beam will spread. Electron beam will so electron beams are converged in particular point. It is going like this and it is going through this crossover point and it is giving us an image at the bottom. Now if we have another possibility that electron beam spreaded like that, it is going through this crossover point and giving us an image. So same thing is true for the black. You can see because of this blue, overall beam diameter is much smaller. For the black it is bigger and the red it is much bigger. Now there is another condition that we have this pink ray. So this is spreaded all over and this is going through like this. When they are going through, we are getting a beam diameter like this. Now in this particular case we are bringing aperture in the picture. So if we have a aperture, so now if you remember that aperture means, I told there is a metal disc, it has some center and rest of the part, these are the metal, it is covered, and whenever ray it will go through this one. So if we put a metal disc like that and from the center ray can go, so it is going to block the electron beam, right. Now for the first condition you consider that I have a aperture size is this. So aperture size, you can get a different type of aperture size. It may be 20 micrometer hole diameter or 30 micrometer or 50 micrometer, 70 micrometer. Those are the hole diameter. Sometimes you will get a 150 micrometer hole diameter also. So here you will get the different sizes hole. You just consider the sum diameter we are using. Now when you are using this sum diameter size, so what is going to happen, it is blocked. So if you just look at this pink, there is a many rays are coming, right, and there are all these pink rays are blocked here. Now some of that red lights are also blocked, and in this particular case if you just consider that red, so this is the ray it is coming and this is the ray it is coming here, and then it will merge and it will give us an image here. So now in this particular case if you look that one, that okay, we have only a aperture diameter is something this big, which is blocking some percentage of the light. If we just consider this some percentage, you can consider that okay, 20% of this light or electron beam is blocked. Now the second possibility that okay, I have this aperture, I want to increase that aperture size. So if I just finish that ray, this ray is going like that, this ray is going, and then I have a red light here. Now I have aperture size is this big. In this particular case you can see that almost 50% electron I am trying to block with this aperture. When I am blocking 50% of the electron, that electron, this black ray, it is coming like this and then it is merging through this particular point. So now you can see that okay, I am just using a very strong spot size or magnetic field, that I am using a very strong magnetic field, meaning that spot size is 8, where the crossover point it will go up. When the electron beam will spread like that, I will use a smaller aperture size and try to block the beam. Here we are blocking almost 50% electron beam. Now if we just use a very short aperture. So, something like that, I will just erase this one. So, if I just use a very short aperture, something like that, I am even trying to block partially this, okay, and allowing only those electrons which are within that range. So, I am just keeping a aperture size something like that, and I am just allowing the electron which are coming within this range. So, someone can say that, okay, so then if we just use the spot size 2, your crossover point, it will be here, and most of the electron will pass through your sample and you will get a very bright beam. But that beam is a very strong, even if you just consider that, okay, whatever electron we are getting, 100 electrons it is coming from this source, or 500 electron it is coming from this source. Now, here if your spot size is this one, you can see even you are using a very small aperture size, even 20 micrometer aperture size, that block majority of the other beam. But if spot size is very small, that is 1 or 2 or 3, you can see most of the electrons are going through this. So, here also your beam diameters is small, and all these 500 electrons are saturated in this particular area, which is not good for your sample. For the biological science sample, biosample, cryo condition, so generally 40 to 80 electron per angstrom square is suitable, okay. So, total electron dose I am talking about. So, if the total electron is this much, that will be suitable. If you are using more, that is going to damage the biological science sample. So, here what I was showing you here, that we have total 500 electrons, and all these 500 electrons is saturated here. So, definitely for the Cryo-EM, this is not the good spot size. So, we need something like that, okay. So, now next condition, if you just consider that when we are increasing the spot size, some of this electron, if you just consider that this is an electron or this is an electron, it is coming like that. So, for higher spot size, even if you just consider that this is a higher spot size, something the spot size 8. So, what is happening, this particular electron beam, if this is the red beam, here you can see this is the ray it is coming, this is the ray it is coming, but all the cases majority of these rays are blocked by this aperture. So, in this particular condition, you can tell that here you have 500 electrons, but due to this aperture almost 80% electrons are blocked. So, finally, if we just use this condition, 80% is blocked, then electrons are coming, only this electron, this much electron can come and give us an image something like that. So, 80% of this one means 100 electron can go through this path and give us an image here. So, in this particular condition, you can see that if you are using a spot size, that higher spot size 8, and then you are using a aperture, smaller size of aperture, and there is a C2 lens current, if you maintain, you can control almost, you can remove almost 80% electron dose, only you can keep 20% electrons, means only 100 electrons you are allowing, and then you can control the beam diameter also. So, your beam diameter is not changing. So, even if you have a cryo grid and if you have a particular hole, you want to control the beam diameter as well as the electron dose. So, you can control your C1 lens current, use a very strong magnetic field, spot size 7, 8 or 9, and then C2 lens current, where you should maintain a proper beam diameter and use an aperture, you can control the electron dose. So then minimum electron can go through your sample. Okay, so now we have to discuss one more thing, that okay, we discussed that how the C1 and C2 we can control. We already discussed that. Okay, this is the electron dose. It is coming in normal way. If we have an aperture, we are blocking most of the beam, so then we are using a higher spot size 8 or 9, then majority of the electron beam will be blocked. Only some electron beam, it will go through the sample, and we can get an image where we can control the electron dose in between 40 to 80 electron dose. If we just consider that 40 to 80 electron dose, we can get it 40 to 80 electron dose, and that will be good for our biological science sample. But one more thing you have to keep it in mind, that last class I discussed that thing. If there is a sample and if electron beams are going, we really don't want electron beam should go and fall on our sample like this. So it should be always parallel. So parallel electron beam, it will come, it will interact with our sample, and then there will be scattering, it will happen. So somehow here you can see there is a crossover point and electron beams are going, the converge and diverge constantly. So somehow we have to control that part, that okay, we have to make this electron, it should not be like this one. So we have to make this electron beam somehow parallel. So there is a two things we have to pay attention here. So now if you just consider that what I was drawing previously, that okay, there is a C1 lens current, they are converged, and then there is a C2 lens. Now just after that there is a upper objective lens and lower objective lens, and then your sample. So someone can tell that okay, what is the best way? So if we just make the beam focused in one particular point, is this a good idea to control your C1 lens current and C2 lens current in such a way that your electron dose, it will go and fall on your sample or focused on one particular position? And so if it will focus in one particular position, it is so much electron dose is concentrated in one particular point, it is going to damage all the amorphous ice and it will melt the amorphous ice. So this is definitely not a good idea. So that is the reason we have to play around with that lens current in such a way with the upper objective lens, so we can get the best possible electron dose as well as the parallel electron beam. How we can get that one? So if we just come again, so now it will be better to draw the optical axis at beginning. So now I am not talking about C1 or C2 changes, because that we already discussed. So this is our optical axis. Right, now from this optical axis, first ray, it will come like that, then there will be a crossover point here. If we are using some strong current, it can go something like that. So here we are not interested about that whatever current, we are maintaining a lens current in such a way that we just interested that okay, some of the electron will be blocked from the black to blue and some of the electron will go. Now we'll use that C2 lens current in such a way that it will not converge in one particular sample. So if this is a sample plane, we will maintain this magnetic field in such a way that ray can go like this. So this is our C2 lens, this is my C1 lens. This is my C1 lens, and this is, I can consider that gun crossover. Okay, and this one is C1 crossover. Okay, so you can see this is the C2 lens. If I just ask you what is the magnetic field for this C2 lens, what kind of magnet, is it a strong, is it a weak, or it is a under focused or over focused? So definitely it is a very weak magnetic field I am using, and that is the reason the electron beam is not converged in any particular point. Not only that, if we just consider this is the rays are going, if you remember under focused image, so how this under focused images are going, if this is my focused image, this is my focused plane, under focused image it will be like that. So you can see, so definitely this lens is under focused condition. So under focused. So now if you just want to measure the angle, this angle is also important, this is the alpha. So we can discuss about those things later stage. So here you can see that okay, we are using a lens current in such a way that C1 and C2, and we are trying to make the beam parallel. It is not perfectly parallel, but it is somewhat, it is parallel, but the beam diameter is very big. So that is also not very good for our purposes. So that is the reason we are going to draw the other one here. So initially I thought I'll make this, okay. So now draw the other one, that will draw a optical axis here. Now use the black ray to draw the lens system. So it is coming like that, and then there is a crossover point. Rays will go in its own way, and now I'll use the other ray. So where this ray will come until here, and then I'm using the C2 lens because here I have a C2 lens. So here that C2 lens I'm trying to focus it, not under focused anymore, because I have a upper objective lens somewhere here. So I want to use that upper objective lens. So I am using it, focused it somewhere here. So there is a crossover point using that C2 lens, and then rays are going like that. Okay, so this is the front focal point. If you remember properly, we discussed about the back focal plane. What is that? The C1 lens, C2 lens, then my sample, it is somewhere here. From the sample, electron beams are coming and converged one particular point, and that is we are recording the diffraction pattern, right? The diffraction pattern when we are recording, that is diffraction pattern when we are recording in this particular point, that is we are calling back focal plane. Now if you just consider the opposite, where the electron beams are converged just above your sample, that is called your front focal point. So if this is a back focal point, the opposite part where electron beams is converged just above your sample, that is called the front focal point. So here this is your C2 lens current. We are using C2 lens current in such a way that electron beam should focus. So electron beam is focused in this particular point. Now we are using the upper objective lens in such a way that upper objective lens can make this electron beam parallel. So if we just make it little bit clean diagram, so diagram it will come look like that. Okay, so this ray it is converged in one particular point, then upper objective lens it is somewhere here. This is our upper objective lens, this is our C2 lens, upper objective lens making this beam parallel. So we are using that upper objective lens and C2 lens together and making the crossover point using the C2 lens in front focal point or front focal plane, which is just above our sample, and try to make the electron beam using the upper objective lens as a parallel electron beam. So this parallel electron beam it will pass through our sample. So here you can see all these lenses has a very important role, where we are using the C1 lens to make the crossover point up, and then we are using C2 lens to converge in one particular point, which is helping the upper objective lens to make the beam parallel, as well as we are using aperture to control the electron beam, how much electron beam is going to pass through the aperture and go through the sample. So we can use different combination of C1, C2 and upper objective lens to control our entire electron beam, whatever it is coming out from the electron gun. So this is the basic ray diagram. I am not going to go in details how to make this entire ray diagram, as well as there are lots of other parameters are also involved. Sometime C3 lens current is also there, which is helping us to make the electron beam parallel, and for 300 kb microscope we are using C3 lens also. So we are not going to discuss about all of these things in details, then it will be very complicated. So we'll just finish this ray diagram part here. In the next class we will discuss about more about that sample holder, because we already discussed the objective lens, upper objective lens, how to make the electron beam parallel. We will talk about the sample holder, and then vacuum systems, and the data collection tool, means basically the different kind of detector, in the next class. Thank you all for your attention.